There are lakes at the bottom of the sea, real lakes, with shores, surfaces, and water so deadly that anything crossing into them dies in seconds. They sit inside the Red Sea, invisible from above, hidden under a mile of ocean. We're going to go down there. We'll find creatures hunting along a poisonous coastline, microbes that might hold the cure for infections killing millions, and a discovery so strange it made scientists question what they thought they knew about life itself. If you enjoy this journey, subscribe and hit like so we can keep going deeper. Brace yourself. We begin. The year was 1972. A research vessel was floating above the central Red Sea, and a scientist was watching a monitor connected to a camera being lowered on a cable into the deep. The camera sank past 100 feet, then 500, then past 1,000 feet, where the light from the surface completely disappears and the ocean turns absolute black. The team had done this dozens of times. They expected to see nothing unusual, just dark water, maybe some sediment drifting past the lens. Then the camera hit something. At least that is what it looked like. The cable kept feeding out, but the image on the monitor showed the camera had passed through a surface, a clear, shimmering boundary like the camera had just entered a different world sitting inside the ocean. Below that boundary, the water looked thicker, heavier, almost syrupy. The scientist stared at the monitor. The camera was still underwater. The cable confirmed it. There was no floor beneath them for another mile. So what had the camera just passed through? It had entered a lake sitting on the bottom of the sea, a body of water resting inside another body of water, with its own distinct surface, its own shoreline, and its own set of rules that had nothing to do with the ocean surrounding it. The team pulled the camera up and checked the water samples collected at that depth. The water was almost 10 times saltier than normal seawater. It was thick with dissolved minerals. It was warm in a way the surrounding deep ocean had no reason to be. And it was clearly completely separate from everything around it. That discovery in the Red Sea changed how scientists thought about the ocean floor. Before that moment, most people imagined the deep sea as one continuous body of water, cold and mixed and uniform. After that moment, the picture got stranger. The ocean floor was hiding lakes, actual lakes with surfaces you could see, shores made of crystal, and water so different from the surrounding sea that the two would barely mix even if you stirred them together for hours. The Red Sea was already famous for being one of the saltiest, hottest, and most extreme bodies of water on the planet. But this discovery suggested something was happening far below its surface that nobody had predicted. Something geological, something ancient, and something that would keep revealing new secrets for the next 50 years and counting. Scientists name these features brine pools. The word brine just means extremely salty water. But calling them pools undersells what they actually are. Some of them stretch for miles, some are hotter than your shower at full blast, some are almost completely airless, and every single one of them is capable of killing almost any creature that enters it within minutes. They exist in other places around the world too, in the Gulf of Mexico, in the Mediterranean, scattered across deep ocean floors on every continent. But the ones in the Red Sea are different, older, stranger, more extreme than almost anywhere else. And the reason they exist at all comes down to something that happened millions of years ago. A geological accident so massive it buried a trap under the entire seabed just waiting to be unlocked. The answer starts with salt, enormous amounts of it, sitting under the Red Sea floor right now. And what happens when seawater finds that salt is where things take a turn no one saw coming. Picture a glass of water. Now imagine dropping a second liquid into that glass, something so heavy and thick that it sinks straight to the bottom and just sits there, refusing to mix with the water above it. The two liquids share the same glass, but they stay completely separate. One on top, one on the bottom, a visible line between them. That is what a brine pool is, except the glass is the ocean. The bottom liquid is water so loaded with dissolved salt and minerals that it behaves more like a liquid metal than regular seawater. and the visible line between them is a shimmering boundary called the halocline. The halocline is the moment the two worlds meet. Cross it going down, and everything changes. The pressure increases sharply. The temperature shifts. The chemistry becomes something most life on Earth simply cannot survive. So why does the dense brine stay put instead of slowly mixing with the ocean above it? Density. That one word explains everything here. Normal seawater has a certain weight per gallon. The brine sitting in these deep pools can be 3 to 8 times heavier per gallon than the ocean above it. Imagine trying to mix honey into a glass of water by just setting the honey on top. Gravity pulls it straight down, and it stays there. The brine does the same thing, except on a scale the size of several football fields. The halocline acts like a physical lid. Water molecules above it stay above it. Water molecules below it stay below it. Currents that sweep through the deep ocean pass right over the top of a brine pool without disturbing what is inside. Storms on the surface thousands of feet above do nothing to the lake below. This means a brine pool is one of the most stable environments on the entire planet. Nothing stirs it. Nothing dilutes it. It just sits there, getting more concentrated over time as minerals continue feeding into it from below. And that stability has a terrifying side effect. Any oxygen that was originally dissolved in the brine gets consumed very quickly by microbes living inside the pool. Once the oxygen is gone, nothing replaces it, because the halocline blocks fresh oxygenated water from entering from above. So most brine pools become completely airless over time. Breathe the water inside one of them and your body gets nothing. This is why fish, shrimp, eels, and most other sea creatures die almost immediately after crossing the halocline. It takes only seconds. Their muscles seize, their cells stop working. They sink to the bottom of the brine lake, preserved almost perfectly in the dense, oxygen-free liquid around them. Scientists have pulled cameras out of brine pools and found them coated with a thick mineral crust after just a few minutes inside. The same chemistry that kills animals fast attacks equipment fast too. The deeper the brine pool, the more intense the conditions, and some of the Red Sea pools sit under nearly a mile of ocean above them, which means the pressure on top of the crushing brine is already enormous before you even factor in what is dissolved inside. Scientists had to engineer special tools just to collect samples from the worst of them, and when they analysed those samples they found something living inside a liquid that should make life completely impossible. That discovery is coming. But first, you need to understand where all this salt came from in the first place. Stand at the shore of the Red Sea on a summer afternoon, and the air temperature can hit 110 degrees Fahrenheit. The water itself, even at the surface, can reach nearly 90 degrees in shallow areas. It is one of the hottest seas on Earth. Now add the evaporation. Because the Red Sea sits in one of the driest regions on the planet, surrounded by desert on both sides, water evaporates off its surface at an enormous rate. Far more water leaves the Red Sea by evaporation than enters it through rainfall. The sea essentially bleeds fresh water into the sky constantly. What gets left behind when water evaporates? Salt, and everything else that was dissolved in it. The Red Sea is about 40% saltier than the average ocean. That matters because saltier water is denser water, and denser water behaves differently. It sinks faster, it circulates differently, it interacts with the seafloor in ways that ordinary ocean water does not. The Red Sea is also one of the narrowest seas connected to a major ocean. It is roughly 1,200 miles long, but only about 200 miles wide at its widest point. Picture it like a long, thin corridor of water, with desert pressing in from both sides, open to the Indian Ocean at one end through a shallow strait called the Bab el-Mandeb. That narrow opening restricts how much fresh ocean water gets in to replace what evaporates, so the Red Sea just keeps concentrating, getting saltier, getting denser, getting more extreme year after year over millions of years. The seafloor under the Red Sea is also unusually young compared to most ocean floors. The rock down there is geologically active, meaning it is still forming, still shifting, still cracking open in places. Heat from deep inside the Earth bleeds up through the thin, young crust more easily here than in older, thicker ocean floors. That heat matters. It drives the chemistry of everything happening in the brine pools below. Warmer rock means warmer water seeping through it. Warmer water dissolves minerals faster. More dissolved minerals mean denser brine. Denser brine means more extreme pools. Every condition the Red Sea has, the heat, the narrow shape, the intense evaporation, the young seafloor, stacks on top of every other condition to create something the ocean does almost nowhere else at this scale, and all of it traces back to a split that started millions of years ago. A crack in the earth so big it created the Red Sea itself. About 5 million years ago, the Red Sea was not a sea at all. It was a desert basin, a low-lying stretch of land between the African and Arabian plates, sitting below sea level but completely dry. The sun hammered it. Nothing lived there. Winds blew across cracked ground. Then the Indian Ocean broke through. A massive flood, far beyond anything in recorded human history, poured over a natural barrier at the southern end of the basin. The water rushed in and filled the depression over thousands of years. The Red Sea was born. But before that happened, during an earlier geological period, a shallow sea had already covered this same area. That ancient sea existed for millions of years and then dried up completely as the plates shifted and the basin was cut off from the ocean. When it dried, it did not leave behind sand, it left behind salt. Enormous amounts of it, layer after layer, baked by the sun over thousands of years into a thick, solid crust of evaporate deposits. Evapораtes are the minerals left behind when water evaporates. Salt is the main one, but there are others too, gypsum, anhydrite, and various compounds that do not dissolve easily once they have crystallized. These mineral layers piled up over time and then got buried when the Red Sea refilled and sediment began accumulating on top of them. So right now, under the Red Sea floor, there are layers of ancient salt sitting hundreds of feet below the modern seafloor. In some areas, these deposits are miles thick. The weight of the ocean and the sediment above them keeps them pressed down, locked in place. Scientists figured this out by studying the seismic waves that move through the seafloor. Sound waves sent downward bounce differently when they hit salt versus rock. The data revealed a picture of the hidden salt layers, and the picture was enormous. The entire central Red Sea sits on top of a salt formation that stretches for hundreds of miles in both directions. It is one of the largest known evaporate deposits anywhere on Earth, and it is not just sitting there doing nothing. It is slowly dissolving from above. Seawater from the modern Red Sea seeps down through cracks and faults in the seafloor, reaches those ancient salt layers, and begins eating through them. The water dissolves the salt, picks up the minerals, and becomes extraordinarily dense brine. That brine then does something simple and inevitable. It falls. It flows down through whatever path it can find, following gravity, and collects in the lowest point it can reach. And the lowest points available are deep depressions on the Red Sea floor. That is where the lakes form. That is where the brine pools sit. The trap was set 5 million years ago when a sea dried up in the desert. The lakes forming today are its delayed consequence. The Red Sea floor is covered in cracks, thousands of them, some small enough to miss, others wide enough to drive a car through. They are the result of the tectonic plates beneath the sea slowly pulling apart, tearing the crust open as they go. These cracks do not stay empty for long. Cold seawater from the ocean above seeps down into them. It moves slowly, pushed by pressure from the weight of the ocean above, filtering down through the fractured rock like water through a sponge. In most parts of the ocean, this would be unremarkable. Water seeps into rock everywhere. Usually it just stays there or gets pushed out somewhere else. Under the Red Sea, the water finds salt. When ordinary water contacts salt, the salt dissolves. This is basic chemistry, the kind you can do in a kitchen. But the scale of what happens under the Red Sea is hard to picture. The seawater is not meeting a spoonful of table salt. It is meeting a layer of ancient evaporate minerals that could be a mile thick, stretching for hundreds of miles. The water dissolves the salt layer from wherever it touches. It picks up sodium, chloride, magnesium, potassium, calcium, and dozens of other minerals in concentrations far beyond anything found in normal ocean water. It becomes brine, a liquid so loaded with dissolved material that it weighs several times more per gallon than the ocean water that created it. The process is slow by human standards. We're talking about water moving through rock at maybe a few inches per year. But geology works on timescales that dwarf human history. Over thousands of years, enormous volumes of this brine accumulate. Once the brine forms, it has nowhere to go but down. Gravity takes over. It seeps through more cracks, moves along fault lines, follows the path of least resistance downward. Wherever the seafloor has a natural low point, a basin or depression created by the tectonic pulling, the brine flows in and collects. This is exactly what happens in your kitchen if you tip a cutting board slightly. Water follows the tilt and pools at the lowest corner. The Red Sea floor is tilted in dozens of places by tectonic activity, and the brine finds every single low point. Once enough brine fills a depression, it creates a stable pool. The pool sits below the halocline. The halocline forms. The lake is born. This process is still happening right now. Active brine seepage is ongoing in multiple areas of the Red Sea. New pools may be forming at this moment in depressions that have not yet been discovered, and the brine that enters these pools does not stay chemically simple. As it moves through different rock layers on its way down, it picks up extra minerals: iron, manganese, zinc, copper. Each pool ends up with a slightly different chemical recipe depending on what kind of rock the brine travelled through to get there. That variation in chemistry is part of what makes the Red Sea brine pools so extreme compared to similar features in other oceans. Because in some of these pools the liquid that collected is not just salty, it is carrying enough dissolved metal to make the water look and behave like something out of a science experiment that went dangerously wrong. The moment enough brine collects in a seafloor depression, something almost unreal happens. The liquid organizes itself, it settles, and it creates a shoreline, a real shoreline, under the ocean. The edge of a brine pool is one of the strangest landscapes on Earth. On one side you have the regular deep ocean, dark and cold and moving with slow currents. On the other side you have the brine, heavier and denser and completely still. Where they meet, minerals crystallize out of the brine and form a white crust along the rim. That crust looks like salt flats you would find in a desert, except it is sitting under a mile of seawater. Scientists who have sent cameras down to these edges describe the halocline as looking like the surface of a completely calm lake on a still day. The ocean above it reflects off the top of the brine below. You can see your own camera reflected back at you from the surface of an underwater lake. That is how clear and distinct the boundary is. If you could stand at the shoreline of a Red Sea brine pool, the scene would feel wrong in every direction. Above you, the ocean stretches upward for nearly a mile. Around you, the rocky seafloor slopes down toward the pool, and at your feet, a shimmering surface separates you from liquid so dense that dropping anything into it causes it to float or sink extremely slowly depending on the object's weight. Objects that would sink in normal seawater sometimes float on brine. The density is that extreme. The size of these pools varies enormously. Some are smaller than a basketball court. Others stretch for more than a mile in length. A few of the Red Sea pools are deep enough that scientists have measured temperature and chemistry changes at different levels inside them, like studying layers of a lake from a boat. The Atlantis Too Deep is one of the largest and most studied brine pools in the Red Sea. It sits about 1,500 miles from the surface of the sea in a depression that formed from tectonic activity. It contains several distinct layers of brine, each one slightly different in chemistry and temperature, stacked on top of each other and barely mixing. The top layer of the Atlantis Too Deep brine is already twice as salty as normal seawater. The bottom layer is more than 7 times saltier. The temperature rises as you go deeper, pushed up by heat coming through the seafloor from the active rift below. The bottom of this pool sits at a temperature hotter than most people set their home ovens, and yet, something lives down there. Scientists sent a sampling device into the Atlantis too deep decades ago, expecting to find nothing alive in such an extreme environment. What came back up changed the conversation about where life can survive on this planet. The year was 2005. A deep-sea research team was lowering a remotely operated vehicle into the Red Sea, following up on reports from earlier expeditions that had detected unusual water conditions in certain areas of the seafloor. The vehicle descended slowly. The team watched a live video feed from cameras mounted on its front. They saw what they expected to see for most of the descent: dark water, the occasional particle drifting past, nothing remarkable. Then the vehicle's depth gauge showed they were approaching a known brine pool area. The team slowed the descent. On the monitor, the water looked slightly different, hazier. The particles in the water seemed to increase. The temperature sensor ticked upwards slightly, then the salinity meter spiked, dramatically. The camera passed through the halocline. The image changed instantly. The water below the halocline looked different. Clearer in some ways, but with a strange shimmer that made it look more like a liquid inside a lava lamp than ordinary water. The vehicle's buoyancy changed too. The brine was so dense that the vehicle, designed to sink in normal seawater, slowed its descent and nearly floated at the halocline before the team adjusted its ballast. The team sent the vehicle forward, skimming just above the brine surface. The camera captured something almost impossible to process visually: a flat, shimmering surface stretching in every direction inside the ocean. A horizon line, a lake with a visible surface sitting under a mile of open water. Then they steered the vehicle toward the shoreline of the pool and saw the crystalline rim. White mineral formations encrusting the edge of the brine like salt on the rim of a margarita glass, except these crystals were the size of softballs. They dipped the vehicle's sampling arm below the halocline and collected a vial of the brine. When they brought it back to the surface and analysed it, the results were striking. The brine contained dissolved minerals at concentrations that would be toxic to virtually every known form of complex life. Iron, manganese, lead, zinc, and dozens of other dissolved metals were present in the sample at levels that would be considered extreme even in an industrial accident. The team marked the coordinates, documented the findings, and submitted their report. But when other scientists read it, several of them had the same question. If the brine is this poisonous to complex life, how was organic material showing up in the samples? Traces of biological activity were in the data. Something was using the brine as a home. The answer to that question would take years to fully work out, and it would eventually push scientists to think about oceans on other worlds. A camera hovering just above the shoreline of a Red Sea brine pool sees a landscape that belongs in a dream sequence. The seafloor leading up to the edge of the pool is covered in a fine grey sediment, flat and featureless. Then the ground starts to change. The sediment becomes whiter. Patches of crystal appear. They grow larger and more dense as the camera moves closer to the edge. Eventually, the entire floor is covered in a crust of white and pale yellow mineral formations, some smooth and flat, others jagged and branching like frozen coral. These formations are called precipitates. When the brine seeps up through the bottom of the pool and contacts the sediment, or when it evaporates very slightly at its own surface, minerals crystallize out of it and harden. Over time, these crusts build up. In some areas, the mineral formations are several feet thick. The edge of the brine surface itself shimmers. The halocline catches the light from the vehicle's floodlights and reflects it back in rippling patterns that look like sunlight hitting a calm river, except there is no sunlight down here. The only light is artificial, and the effect is still striking enough that the scientists watching the feed often go quiet. Just past the rim, the surface of the brine stretches out flat and still, almost mirror-like. The halocline is so stable in most of these pools that the brine surface barely moves even when a remotely operated vehicle passes inches above it. The currents of the deep ocean, which are slow to begin with, do not penetrate below the halocline. The colours change as the camera goes deeper. Near the rim, the brine can be pale and nearly clear, even though it is already many times saltier than the ocean above. Deeper into some pools, the water takes on different colours depending on what minerals are dissolved in it. Some pools have a rusty orange tint from dissolved iron. Others have a yellowish cast from sulphur compounds. A few have a dark metallic sheen near the bottom where heavy metals concentrate. The seafloor inside the pool itself is different from the floor outside. The sediment inside a brine pool accumulates in completely undisturbed layers because nothing stirs the brine from inside or outside. No currents, no burrowing animals, no bioturbation, which is the scientific word for animals mixing the sediment up as they move through it. So the floor inside a brine pool is like a perfect archive. Every layer of sediment preserves exactly what fell into the pool at the time it was deposited. Scientists can read these layers like tree rings, moving backward through time with each inch they drill down. Some of these archives go back hundreds of thousands of years. What they contain is remarkable, and what scientists found at one particular depth, in one particular layer, raised a question nobody had thought to ask before about what was living in this region long before humans showed up. A shrimp drifts too close to the edge. Maybe a current carries it. Maybe it is chasing food. Whatever the reason, it crosses the halocline. The moment it does, the chemistry around it shifts completely. The shrimp's body is built for normal seawater. Every cell in its body is in a careful chemical balance with the water around it. Cells take in what they need and push out what they do not, through a process called osmosis. This process depends on the difference in salt concentration between the inside of the cell and the outside. In normal seawater, that difference is manageable. The shrimp's body handles it without effort. Inside the brine, the salt concentration outside the shrimp cells explodes. It becomes so much higher than the concentration inside the cells that water is pulled out of the cells by osmotic force, rapidly and violently. The cells begin to shrink and collapse. Every tissue in the shrimp's body starts to fail at once. The shrimp cannot breathe either. The brine has almost no dissolved oxygen. The gill structures that extract oxygen from water find nothing to extract. The animal suffocates and dehydrates simultaneously, from the inside out and the outside in. This happens in seconds, fast enough that the animal has no time to escape, even if it was somehow aware of what was happening. The shrimp sinks to the brine floor, and here is the strange part. It does not decompose the way a dead animal normally would in the ocean. Normal ocean water carries bacteria, oxygen, and organisms that break down dead matter. The brine carries almost none of those things. The near-total absence of oxygen means aerobic bacteria cannot survive in the brine to decompose the body. The density of the liquid preserves the shrimp's shape. The chemicals in the brine actually begin to fix the body's tissues, similar to how a preservative works. Scientists have found dead fish, crabs, eels, and shrimp at the bottoms and along the edges of brine pools in conditions of preservation that are remarkable. Animals that died weeks or months ago still have intact scales, intact limbs, clear eye structures. Some look freshly dead. This is the brine acting as a natural formalin. The same toxic chemistry that kills the animal begins preserving it the moment it dies. This preservation effect has enormous value for paleontology and geology. Anything that fell into a brine pool over the past few thousand years is likely still down there, essentially intact, waiting to be studied. Shells, bones, ancient wood from ships, organic material from organisms that no longer exist in the Red Sea. All of it can be sitting in perfect condition on the floor of a brine lake right now. The creatures that live along the shoreline of these pools have figured out how to use this death trap to their advantage. They wait at the edge, and when prey stumbles too close, the brine does the work for them. The rim of a brine pool is one of the most productive hunting zones in the deep sea. Thick mats of bacteria grow along the halocline and across the mineral crust at the shoreline. These mats are dense enough to look like orange and white carpets from a camera above. They attract tiny animals that feed on bacteria. Those tiny animals attract slightly larger predators, and those predators attract larger ones still. A full food chain operates right at the edge of a lake that would kill most of the animals in that chain if they crossed 1 inch too far. Eels are some of the most dramatic hunters along brine pool edges. The barbelled dragonfish and certain species of deep-sea eel have been filmed hunting along haloclines in the Gulf of Mexico and similar brine environments. The strategy is simple and brutal. The eel hovers just above the halocline surface and waits. When a fish or shrimp crosses the boundary and becomes paralysed or dies from the brine shock, the eel dips below the surface just far enough to grab it, then pulls back up into normal seawater. The eel is essentially using the brine like a paralysing net. Deep-sea shrimp gather in enormous numbers around the bacterial mats at the edge of Red Sea brine pools. Scientists observing these areas with cameras have counted thousands of shrimp in a single frame, crowded along the shoreline like fans pressing against a concert barrier. The shrimp are careful. They feed on the bacterial mats without crossing into the brine. When the camera moves too close, they scatter back from the edge, never toward it. Polychaete worms, which look like hairy centipedes and can reach over a foot in length in the deep sea, build tube structures in the sediment right at the halocline boundary. They extend feeding tentacles into the bacterial mat and pull back when anything disturbs them. Some of the strangest observations come from the bacterial mats themselves. The mats are thick with millions of microbes, but they are organised. Different species occupy different zones within the mat. Some live in the oxygen-rich water just above the halocline. Others live in the transitional zone right at the boundary. A small number live in the brine itself. The ones living inside the brine are doing something that should not be possible. They are not just surviving in a liquid that kills everything else. They are thriving, reproducing, processing chemicals that no other life on Earth relies on for energy. These microbes are the real discovery, and what scientists found when they studied them changed the idea of what life actually requires to function. The floor of a brine pool is a museum. Nothing disturbs it. No currents stir the sediment. No burrowing animals tunnel through it. No bacteria break down what lands on it. Whatever falls into a brine pool is sealed into the sediment in the order it arrived, layer by layer, year by year, for thousands of years. Scientists call this kind of undisturbed layering a laminated sediment record. It is extraordinarily rare in ocean environments. Normally the seafloor is constantly churned by biological activity. Worms burrow through it. Fish root around in it. Bacteria consume organic material and release gases that bubble up through the layers. All of this mixing destroys the record. Inside a brine pool, none of that happens. A team of scientists drilled a core sample from the floor of a Red Sea brine pool called the Kibrit Deep, which sits at a depth of around 5,000 feet below the sea surface. When they pulled the core up and sliced it open, they found layers going back approximately 20,000 years, each one distinct and readable. They could see the layer from the last ice age. They could see chemical shifts that matched known climate events from thousands of years ago. They could identify periods of increased volcanic activity from tiny ash particles trapped in the layers. And they found organic material, actual biological molecules, still chemically intact despite being buried for millennia. Preserved organic molecules from thousands of years ago can tell scientists what organisms were living in the Red Sea at different periods in history. Some of those organisms no longer exist. Some reveal ancient conditions of the sea that written history does not capture. One layer in the Kibrit Deep core contained traces of organic compounds linked to a mass die-off event in the surrounding sea. The chemical signature pointed to a dramatic change in salinity or temperature that overwhelmed the ecosystem. The exact cause is still being debated, but the brine preserved it. While the rest of the seafloor lost that record to biological activity thousands of years ago, the brine pool held it perfectly. The bodies of dead animals found in brine pools add to this archive, A fish that died decades ago sitting on a brine pool floor may have its stomach contents still intact. What it ate, where it came from, what chemicals were in its tissue at the time of death, all preserved in a level of detail that normal ocean sediment cannot offer. Scientists have been pushing to map and sample every accessible brine pool in the Red Sea before any human industrial activity reaches them. Because once the floor is disturbed, the archive is gone. Some of these pools are sitting above something even more striking than ancient organic material. The deep cores from a few Red Sea brine pools have revealed layers that do not match anything in the documented climate record of the region. They point to events older than the Red Sea itself. At the edges of the Red Sea and in shallow water areas where ancient brine seeps once existed, archaeologists and geologists have found objects that survived intact in ways that defy the normal rate of decay. Wood fragments from ancient vessels, rope fibres, organic material from human activity going back thousands of years, preserved in conditions that suggest contact with brine-rich environments protected them from the decomposition that would have destroyed them in normal seawater. The Red Sea was one of the most travelled trade routes in the ancient world. Ships from Egypt, Arabia, East Africa, and India crossed it for thousands of years. Many of those ships sank. Most of the wooden hulls rotted away centuries ago, leaving only pottery shards and metal fittings for archaeologists to find. But in areas where brine seepage altered the chemistry of the sediment, some organic material survived. Scientists studying one excavated site on the Egyptian coast found rope fibres estimated to be over 4,000 years old, preserved well enough to identify the plant material used to make them. Down in the deeper brine pools, the conditions are even more preserving. The near-complete absence of oxygen and the high mineral content in the brine create an environment where organic breakdown slows to almost nothing. A research team exploring the floor of one Red Sea brine pool found a section of seafloor covered in what appeared to be chunks of ancient wood. Dating analysis suggested the wood was several thousand years old. Its structure was still intact enough to identify the tree species. What a ship's cargo, biological samples or human remains could reveal, if found in similar condition inside a deep brine pool, has pushed several research groups to propose targeted searches. The Red Sea's history as a trade route makes the odds of finding preserved artifacts in brine pools significantly higher than in most ocean environments. The chemistry that makes brine pools lethal also makes them archival vaults. But the archive is only part of what makes the Red Sea floor unusual. The geology underneath these pools is doing something active, something that has been building energy for millions of years, and what is happening beneath the brine is connected to a force big enough to eventually split an entire continent in two. Africa and Arabia used to be one landmass. They sat fused together for hundreds of millions of years. Then, roughly 30 million years ago, the tectonic plates beneath them started pulling apart. Slowly at first, just a crack, a weakness in the crust. But the forces involved were enormous, driven by convection currents deep inside the Earth that pull plates apart the way heat pulls the top layer off a pot of soup. The crack widened. Over millions of years, the land between the two plates sank and stretched. Eventually, the low point of the rift filled with seawater, and the Red Sea was born. The same process created the Gulf of Aden and the East African Rift Valley, which contains the deepest lakes in Africa, and is the beginning of a rift that will eventually split the African continent apart millions of years from now. The Red Sea Rift is still active. The two plates are moving apart at roughly an inch per year. That sounds slow, but over millions of years it adds up to the distance between continents. In a few million years, the Red Sea will be significantly wider. Eventually, it will look like the Atlantic Ocean looks today, wide and deep and fully developed. Right now, the rift is in a young and active stage. The seafloor in the central Red Sea is still forming. Hot material from the Earth's mantle rises up through the rift to create new crust. As it does, it brings heat to the seafloor. That heat drives everything. It warms the water seeping through the cracks in the seafloor. It accelerates the dissolution of the ancient salt layers below. It feeds the hydrothermal systems that pump mineral-rich water into the brine pools. In the deepest parts of the Red Sea, you can find features called hydrothermal vents, cracks in the seafloor where superheated water jets out into the ocean. These vents exist because of the rift, and in the Red Sea, some of those vents are located directly in or near the brine pool depressions. When a hydrothermal vent pumps into a brine pool, the chemistry of the pool changes dramatically. The temperature rises, new minerals enter the liquid, the conditions inside the pool become more extreme than brine chemistry alone can produce. Some of the pools near active vents are hotter than boiling water, and they stay liquid. A hydrothermal vent is a crack in the seafloor where water, superheated by the rock below, pushes back up into the ocean. Imagine a kettle boiling deep underground, with the steam finding a way out through a crack. That is roughly the picture. In the Red Sea, some of these vents sit directly inside or at the edges of brine pool depressions. When they fire, they do not push hot water into the cold ocean. They push hot water into the already extreme brine. The result is a pool that behaves like nothing else on Earth's surface. The vent water carries minerals dissolved from the hot rock below. Iron, zinc, copper, manganese, and heavy metals come up with the water and enter the brine. Over thousands of years, this creates a soup of dissolved metals that concentrates in the pool the same way the salt and minerals concentrated from the ancient evaporate layers. The Atlantis 2 Deep is one of the best-studied examples of a vent-fed brine pool in the Red Sea. Scientists measuring its temperature found that the lower layer of the brine sits at around 60 degrees Celsius, which is 140 degrees Fahrenheit. That is hot enough to sterilize a wound, hot enough to kill most known organisms instantly on contact, and that temperature is stable year-round, driven by the consistent heat output of the vent below. The brine in this pool also carries some of the highest concentrations of dissolved heavy metals ever recorded in a natural water body. Zinc concentrations hundreds of times higher than normal seawater. Iron at levels that turn portions of the water a rust colour visible in camera footage. When scientists first analysed samples from this pool, they debated whether the metal concentrations were a measurement error. They ran the analysis again. Same result. The water inside some of these vent-fed pools is more concentrated with dissolved metals than many industrial wastewater streams, and the heating from the vent creates circulation patterns within the pool that stir those metals into the different brine layers. The pool is not static, it is slowly churning, very slowly, driven by the temperature difference between the hot bottom and the cooler upper brine. This circulation matters for the life inside the pool, because those minerals, even in concentrations that would kill most animals, are fuel for certain microbes. The microbes use chemical energy from the minerals the same way plants use energy from sunlight. They do not need sunlight, they do not need oxygen, they run entirely on chemistry, and they thrive in conditions that look, to every human standard, like a death chamber. Here is something that should be impossible. Water boils at 212 degrees Fahrenheit at sea level. That is the limit. Once water reaches that temperature at normal atmospheric pressure, it turns to steam. It cannot stay liquid. But at the bottom of the Red Sea, inside certain vent-fed brine pools, temperatures have been measured that far exceed boiling, and the water stays liquid. The reason comes down to pressure. At a depth of several thousand feet below the ocean's surface, the weight of all that water above presses down with enormous force. That pressure raises the boiling point of water significantly. The deeper you go, the higher the boiling point rises. At the depths where the Red Sea brine pools sit, water can remain liquid at temperatures that would instantly vaporize it at the surface. So when vent water, at 250 or even 300 degrees Fahrenheit enters a deep brine pool, it does not boil the pool, it heats it. And the brine, already denser and more stable than normal water, holds that heat. The result is a pool of liquid that is hotter than your kitchen oven, sitting under a mile of ocean, staying perfectly liquid because the pressure above it will not let it turn to gas. The boundary between the boiling hot brine and the regular deep ocean above it is striking even in camera footage. The halocline in these pools shimmers more intensely than in cooler pools. Rising heat from the brine creates small convection patterns just below the surface. The halocline almost pulses. Scientists studying one particularly hot Red Sea brine pool found that the temperature gradient between the bottom of the pool and the top of the brine was steep enough to create distinct thermal layers. The bottom layer right above the vent was nearly too hot to measure accurately with standard equipment. The top layer was significantly cooler, but still far warmer than the surrounding deep ocean. Life survived in all of those layers. Microbes collected from the coolest upper layer of one of these hot brine pools were already extreme compared to normal ocean bacteria. Their cell membranes were chemically different, adapted to remain stable in high temperatures and high salt concentrations. Their enzymes worked at temperatures that would destroy the proteins of most living organisms. The microbes from deeper layers were even more adapted, and the deepest samples taken from just above the vent openings themselves contained organisms that broke records. Scientists call them extremophiles. That word means organisms that love extreme conditions, but it undersells what the microbes living inside Red Sea brine pools actually do. Loving an extreme condition usually means surviving it. These microbes are doing something further. They're using those conditions as raw materials. Pull a sample from inside a Red Sea brine pool and bring it up to a lab. Analyze what is alive in it. What you find is a community of single-celled organisms, mostly from the domain of life called archaea, and certain extreme bacteria that have engineered themselves at the molecular level for an environment that was never supposed to have life in it. Their cell membranes are built from different chemical compounds than those of normal bacteria. Regular bacterial cell membranes fall apart in high salt concentrations. The membranes of brine-adapted archaea stay stable. Their structure is fundamentally different, built from chemical bonds that high salt cannot break. Their enzymes, the proteins that run every chemical reaction in their cells, are also rebuilt for this environment. Normal enzymes fall apart above a certain temperature, like cooking an egg. The proteins denature, lose their shape, stop working. Brine pool archaea have enzymes built from amino acids arranged specifically to keep their shape at temperatures that would cook everything else, and they do not use oxygen for energy. The brine pools are nearly airless. Instead, these microbes use chemical reactions between sulfur compounds, iron, and other minerals dissolved in the brine. They strip electrons from one molecule and pass them to another, running a chemical current through their cells the same way a battery works. This is called chemosynthesis. It is the biological equivalent of powering your house with chemistry, instead of electricity from the grid. The microbes generate all the energy they need from reactions between dissolved minerals. The implication of this is enormous. These microbes do not need the sun. They do not need oxygen. They do not need temperature ranges that humans or fish or plants need. They need minerals. They need water, even extreme water. And they need time. A team studying the brine pools of the Red Sea found archaea in samples from pools where the salt content was 6 times higher than normal seawater. The temperature exceeded 140 degrees Fahrenheit, and the oxygen level was essentially zero. The microbes were present in those samples at densities of millions of cells per milliliter of brine. Not surviving, thriving. The discovery shifted the scientific conversation about life in the extreme ocean, and it pointed outward, toward other places in the solar system that share similar conditions. Europa is one of Jupiter's moons. It is covered in ice. Under that ice, scientists are confident there is a liquid water ocean, kept warm by gravitational forces from Jupiter squeezing and stretching the moon like a rubber ball, generating internal heat. That ocean is in total darkness. The ice above it blocks all sunlight. The seafloor below it is in contact with a rocky core that generates heat and minerals. The water is salty. The pressure at depth would be extreme. Sound familiar? The conditions in Europa's subsurface ocean match the conditions inside a Red Sea brine pool in several key ways. Darkness, extreme chemistry, possible mineral-rich water, heat from below. Enceladus is one of Saturn's moons. It actively vents water from its interior through cracks in its icy surface, spraying plumes into space. Those plumes contain water, salts, organic molecules, and silica particles, all consistent with hydrothermal vent activity at the bottom of a subsurface ocean. Scientists analysing the plumes found hydrogen, a direct byproduct of the chemical reactions that deep-sea microbes on Earth use for energy. Enceladus may have hydrothermal vents on its seafloor, producing exactly the kind of mineral-rich, superheated water that feeds the Red Sea brine pools. The microbes in those pools prove that chemosynthesis, running life on chemistry instead of sunlight, can support entire ecosystems in complete darkness, with no oxygen and extreme chemistry. If that works here on Earth, the same process could work on Europa or Enceladus. The Red Sea brine pools became a scientific model case, a way of testing, right here on Earth, what life in an alien ocean might look like and what tools would be needed to detect it. Scientists now study the brine pool microbes specifically to understand their biosignatures, the chemical traces that living things leave behind. If a spacecraft ever collects a sample from Europa's ocean or Enceladus's plumes, scientists need to know what biological evidence looks like in extreme brine-like conditions. The Red Sea is giving them the reference data. A mission to Enceladus is already in planning stages. Europa Clipper, a spacecraft built to study Europa's ocean from orbit, launched in 2024. The data it collects will be compared against what scientists already learned from places like the Atlantis Too Deep. The lakes at the bottom of the Red Sea may be the closest thing to an alien ocean humanity has ever been able to study with physical samples, and what those samples contained was so unexpected that it opened a door into medicine that nobody saw coming. 4 billion years ago, Earth had no oxygen, no ozone layer, no forests, no fish. The surface was bombarded by radiation, hit by asteroids, and covered in young volcanic rock. The oceans were hot, acidic in places, and loaded with dissolved minerals. Life started anyway. Scientists have spent decades trying to figure out exactly where and how. One leading idea points to hydrothermal vents on the ancient ocean floor, places where mineral-rich hot water met cold ocean water, creating chemical gradients that could drive the first biological reactions. The chemistry of those ancient vents, scientists believe, looked a lot like what is happening inside Red Sea brine pools right now. The minerals inside the brine pools, iron, sulfur, magnesium, zinc, and manganese, are the same minerals believed to have been present in the early ocean. These elements are central to the chemistry of the most ancient biological processes known to science. The enzymes at the core of cellular respiration, the very machinery that powers every living cell, still use iron and sulfur at their active centers. These elements were not chosen randomly by evolution. They were available in the early ocean, and life built itself around them. When scientists study the chemical reactions happening inside Red Sea brine pools, they find processes that mirror what they believe happened in the first cells. Sulfur compounds reacting with iron, hydrogen being produced and consumed, simple organic molecules assembling from mineral reactions. A research team analysing brine pool sediment layers found amino acids, the building blocks of proteins, forming spontaneously from chemical reactions between brine minerals and organic compounds in the sediment. These were abiotic amino acids, meaning life did not make them. The chemistry of the brine made them on its own. This is significant, because if brine chemistry can produce amino acids without biology, it suggests that environments like these could have been where biology first got its raw materials. The brine pool becomes a natural laboratory, running the same experiment Earth ran 4 billion years ago, over and over, in slow motion. The extreme conditions in the pools also help test the limits of what molecules can survive. Scientists studying which organic compounds remain stable inside the brine and which break down are building a map of what early life would have had to work with and what it would have had to avoid. Some of those stable compounds are remarkably complex. far more complex than simple salts or minerals. Their presence in the brine pool sediment points to a chemistry rich enough to have supported the first biochemical reactions that eventually became living cells. The Red Sea brine pools are running a 4-billion-year-old experiment in a bottle, and they might be giving scientists the clearest window yet into the moment life crossed from chemistry into biology. But there is a more immediate application to what scientists found inside these pools, one that is already being developed into treatments, and it starts with a microbe that produces something no antibiotic currently on the market can match. Every year, more than 1 million people die from infections caused by bacteria that no longer respond to antibiotics. Drug-resistant infections are one of the fastest-growing medical crises in the world. Bacteria evolve. Antibiotics that worked 20 years ago sometimes do nothing today. The standard approach to finding new antibiotics has been to search soil, plants, and ordinary ocean environments for microbes producing compounds that kill bacteria. Most of those environments have already been searched extensively. The compounds coming from them are increasingly familiar to resistant bacteria. Scientists working on the Red Sea brine pool started asking a different question. What if you searched an environment so extreme that the bacteria there had never been exposed to common antibiotics? What if isolation in a brine pool for thousands of years forced microbes to develop entirely new chemical weapons to fight each other? Compounds that resistant bacteria had never encountered before. The logic was sound. Bacteria in normal environments have had millions of years of exposure to the chemical warfare that happens between competing microbial species. Resistant strains evolved partly by encountering those compounds over and over, but a microbe sealed in a brine pool with no connection to the outside world would be running a separate evolutionary experiment. The chemicals it produced to compete with its neighbours would be unfamiliar to anything outside that pool. A research team collected samples from multiple Red Sea brine pool environments, targeting the microbial communities in the brine and the sediment just inside the halocline. They cultured the microbes in the lab and screened the compounds they produced for biological activity against known drug-resistant bacteria. The results were striking. Several compounds isolated from Red Sea brine pool bacteria showed strong activity against bacteria resistant to multiple existing antibiotics, including strains of drug-resistant staph infections and gram-negative bacteria that current drugs struggle to affect. One compound in particular, produced by an archaea species from a deep Red Sea pool, showed a mechanism of action completely different from any existing antibiotic class. It targeted a part of the bacterial cell wall that current drugs do not touch. Resistant bacteria had no existing defense against it. The research is still in early stages. Moving from a promising lab result to an approved treatment takes years of testing and development. But the discovery established something important. The brine pools are a biological library of novel chemistry, sealed away from the outside world for long enough that the compounds inside are genuinely new to medicine. Other research groups have since reported similar findings from brine environments in the Mediterranean and the Gulf of Mexico. But the Red Sea pools, because of their age, depth, and the extreme chemistry that isolated their microbial communities for so long, remain the richest source discovered so far. The same isolation that made these pools deadly to most life made them a vault of biological innovation. The moment research groups published findings about novel antimicrobial compounds from Red Sea brine microbes, the attention shifted fast. Pharmaceutical companies had been watching the antibiotic resistance crisis for years. Their own pipelines of new antibiotic development had slowed dramatically by the early 2000s. Finding genuinely new classes of antibiotics had become one of the hardest problems in medicine. The brine pool findings represented exactly what the industry had been looking for, a source of biological chemistry that had not been tapped before. Several biotech companies began partnering with research institutions to gain access to brine pool samples. The logistics are not simple. Collecting material from pools sitting under thousands of feet of seawater requires deep sea vehicles, specialized sampling equipment, and permits from the nations with territorial claims over the Red Sea. Saudi Arabia and Egypt share jurisdiction over the central Red Sea, where most of the major brine pools are located. Both governments have set up research frameworks for accessing the seafloor. Several joint research agreements have been signed between Middle Eastern universities, European research institutes, and biotech companies specifically targeting brine pool biology. The process of screening brine microbes for useful compounds is also technically demanding. Many of the organisms living in the brine pools cannot survive in normal lab conditions. Scientists have had to develop pressurized, high-salt, low-oxygen lab environments just to keep the organisms alive long enough to study them. Some brine pool microbes die within hours of leaving their native conditions, even when researchers try to replicate the brine chemistry precisely. Getting around this problem has pushed advances in lab technology. New techniques for extracting and analysing the genetic material of microbes without culturing them in the traditional sense, called metagenomics, have allowed scientists to read the full catalogue of potential compounds a microbe could produce, even without keeping the organism alive in the lab. The genes for producing specific chemical compounds can be identified in the genetic sequence, and then transferred to other, more lab-friendly organisms to produce the compound at scale. This approach has opened up the brine pool microbe library in a way that traditional culturing never could. Instead of being limited to the organisms scientists can keep alive in a lab dish, they can now access the full chemical potential of every organism in the pool through its genetic code. The race to catalogue and patent compounds from brine pool organisms is already underway. Scientists have noted that the window for discovery is tied directly to how well these environments are protected. Any large-scale industrial activity on the Red Sea floor risks altering or destroying the brine pool ecosystems before they can be fully inventoried, and there is pressure on the seafloor that has nothing to do with medicine. Scientists mapping the central Red Sea in detail did not find one brine pool. They found dozens, and in several locations the pools were stacked. Multiple distinct brine bodies sitting at different depths within the same basin, separated by layers of sediment or slightly different water, each one a separate chemical environment. The Atlantis 2 Deep, one of the largest and most studied Red Sea brine systems, contains at least two major brine layers, the upper and lower convective layers, separated by a zone of transition. But the area around it contains additional pools at nearby depressions. A complete map of the region looks less like a single lake and more like a system of lakes, interconnected through the geology of the rift but chemically distinct from each other. The discovery of stacked pools raised new questions. If brine forms by seawater, dissolving ancient salt layers and sinking, how does it end up at multiple depths? Part of the answer is timing. Different episodes of brine formation happened at different points in the geological history of the region. Older brine that formed thousands of years ago settled at the deepest available depression. Newer brine, forming more recently, settled at shallower depressions or on top of existing pools, separated by a density gap. Some pools interact with each other through the sediment between them. Brine from a deeper pool can slowly migrate upward through the sediment and enter a shallower pool, mixing their chemistries gradually. Scientists have mapped these interaction zones using chemical tracers, molecules that appear in one pool and show up in a neighbouring pool at a later sampling date. The stacked pool systems also create layered ecosystems. Each brine layer has its own microbial community, adapted to its specific temperature, salinity, and mineral chemistry. Moving from one layer to another, even a short distance in terms of depth, means crossing into a completely different biological world. A sampling device lowered through multiple layers in a stacked system collects organisms that are evolutionary strangers to each other, despite living only a few feet apart. The haloclines between them are barriers as real as walls. Scientists studying the vertical structure of these stacked systems found that the top layer of a brine pool is often the most diverse in terms of microbial species. It is the layer closest to the ocean above, where small amounts of oxygenated water occasionally seep across the halocline. That tiny trickle of oxygen supports species that cannot survive deeper down. The deeper layers become progressively more specialised. By the time you reach the bottom of the deepest layer in a stacked system, the microbes living there are often unique to that specific pool, found nowhere else on Earth, which meant every pool scientists opened was potentially a door into a completely original biology. 2 brine pools can sit just a few miles apart on the Red Sea floor and have almost nothing in common chemically. One pool might be rich in iron, with water that has an orange tint from dissolved ferrous compounds. Another a few miles away might be dominated by hydrogen sulfide, with a sharp chemical smell that equipment sensors can detect even at depth. A third might be nearly free of heavy metals but extremely high in magnesium, with a chemistry closer to certain industrial fluids than to seawater. The variation comes from the path each brine took on its way to the pool. Brine forms when seawater dissolves through ancient salt deposits. But those salt deposits are not chemically uniform across the entire Red Sea floor. Different sections of the evaporate layer have different compositions, reflecting the different minerals present in the water when the ancient sea dried up millions of years ago. Some sections are rich in chloride salts, others have more sulfate minerals, others contain pockets of metallic ore that form when hydrothermal activity concentrates metals in rock over geological time. When seawater dissolves through a sulfate-rich section of the evaporate layer, the brine it creates is sulfate-rich. When it passes through a zone of metallic ore, the brine picks up those metals. The pool it eventually fills reflects exactly what the brine dissolved on its way there. Some pools are also geothermally heated more than others, depending on how close they sit to an active part of the rift. Higher heat accelerates chemical reactions inside the pool, concentrating some compounds and breaking down others. A pool sitting directly above an active vent has a completely different interior chemistry than a pool a mile away from the nearest thermal source. Scientists have documented pools with acidic conditions where the water eats through metal sampling equipment quickly, others where the chemistry is nearly neutral, pools where the oxygen is completely absent, pools where certain gases like methane or hydrogen sulfide bubble up from the sediment in quantities large enough to make sampling complicated. Each pool is its own chemical planet. This variety matters beyond scientific curiosity. Each chemically distinct pool hosts a distinct microbial community. Those communities have adapted to their specific pool's chemistry and no other. A microbe from an iron-rich pool cannot survive in a sulfate-dominated pool. A microbe from a high-temperature vent pool cannot survive in a cooler, metal-free pool. Every pool is a separate evolutionary experiment, sealed off from the others by the halocline and the seafloor between them, running for thousands of years. The tools scientists needed to read all of these experiments had to get a lot more advanced before the full picture started coming together. Getting a camera into a brine pool is one challenge. Getting detailed measurements from the floor of a pool hundreds of feet deep, in liquid that destroys electronics and coats instruments in mineral crust within minutes, is an engineering problem in a different category. Early exploration of the Red Sea brine pools used simple lowered instruments, bottles that would open at a set depth to collect samples, and basic temperature and salinity sensors. These gave scientists enough information to know the pools were extraordinary, but almost nothing about what was at the bottom of the pools themselves. The shift came with remotely operated vehicles, robots controlled from surface ships through cables that can transmit power and video in real time. Modern deep-sea vehicles are built from titanium and engineered polymers that resist corrosion. Their cameras are sealed in housings built to survive crushing pressure. Their sample collection arms can reach out, scoop sediment from a seafloor, and seal it in a container without exposing it to the surrounding water. Even so, brine pools push these vehicles to their limits. The halocline is a physical shock to any vehicle designed to move through uniform seawater. The density jump at the halocline boundary changes buoyancy instantly. A vehicle calibrated to hover in normal seawater will begin to float at the halocline when it tries to enter the brine. Pilots have to compensate by increasing the vehicle's downward thrust significantly just across the boundary. Once inside the brine, the vehicle's thrusters have to work harder to move because the liquid is denser and more resistant to movement. Navigation becomes more difficult because the instruments designed to measure position and orientation were calibrated for normal seawater. A team from a European Oceanographic Institute deployed a specially modified vehicle into the Atlantis Too Deep to map the pool floor in detail. They equipped the vehicle with sonar systems that could build a three-dimensional map of the pool floor even through the optically challenging brine. Over several dives spanning multiple research cruises, they assembled a detailed map of the basin floor, its depth variations, the locations of vent openings, and the distribution of sediment types across the floor. What the maps revealed surprised even scientists who had been studying these pools for years. The floors of several Red Sea brine pools are not flat. They have ridges, valleys, and depressions within the depression itself, shaped by ongoing vent activity and sediment accumulation over thousands of years. One pool's floor showed clear evidence of a relatively recent collapse event, where a section of sediment had slumped inward, exposing deeper layers that would normally be buried. That exposed cross-section gave scientists a vertical view through thousands of years of sediment history in a single image. At the very bottom of the deepest sections of these pools, the data showed something unexpected about the mineral composition of the floor. It suggested a much more ancient origin for some of what was sitting down there than the pool itself. Every particle that falls into a brine pool stays exactly where it lands. That is the key fact. In normal ocean sediment, a particle settles on the floor, and within weeks or months, some organism has burrowed through the layer around it. Some current has shifted it slightly. Some bacterial process has begun breaking it down. The record gets scrambled. Inside a brine pool, none of those processes operate. The lack of oxygen prevents aerobic bacteria from decomposing organic material. The density and stillness of the brine prevents currents from moving particles. The near-total absence of macroscopic animals means nothing burrows, nothing feeds on the sediment, nothing moves through it after it settles. A particle that falls into a Red Sea brine pool lands on the floor and stays in that exact position for thousands of years. Scientists call this kind of sediment record a perfectly laminated archive. Each layer represents a specific window of time. The layers are thin, sometimes only a fraction of an inch thick per century, but they are distinct and readable. A sediment core drilled from the floor of the Kibrit Deep Brine Pool and analysed in detail revealed layers going back more than 20,000 years. Scientists could identify the layer from the peak of the last ice age, when global temperatures were significantly colder, and the Red Sea itself was shallower and saltier than today. They could see a layer corresponding to a period of increased aridity in the surrounding region, when dust from the Arabian and African deserts blew into the sea in larger quantities. They found a layer with elevated concentrations of certain isotopes that matched a known period of volcanic activity in the region. The ash and chemical signatures from that eruption, thousands of miles away, had drifted into the Red Sea and settled into the brine pool, where they sat undisturbed for thousands of years waiting to be read. Biological material in the layers tells its own story. Microscopic shells of ancient marine organisms called foraminifera accumulate in the brine pool sediment and preserve the chemistry of the water they grew in. By analysing the isotope ratios in those shells, scientists can reconstruct the temperature and salinity of the Red Sea surface at the time those organisms were alive. This is how scientists build climate records that go back further than any written history. The Red Sea brine pools have provided climate data that is cleaner and more detailed than almost any equivalent core from a normal seafloor, simply because the brine protects everything that enters it from degradation. A few of those cores have revealed layers that do not fit any known climate event in the Red Sea's recorded history. They point to something older and larger. A sediment core is a cylinder of compressed history. Scientists lower a hollow metal tube into the seafloor and push it in with controlled force. When it comes back up, the tube contains a column of sediment with the oldest material at the bottom and the youngest at the top. If the sediment has been undisturbed, the layers inside the core are pristine. Each inch represents a specific slice of geological time. The cores pulled from Red Sea brine pool floors are among the most detailed sediment records from any ocean on Earth. In a core from the Atlantis Too Deep region, scientists found layers that extended the record deeper than the typical brine pool sediment archive. Below the layers corresponding to known climate events of the last few thousand years, the core showed a distinct chemical boundary. Above it, the sediment had the chemical signature of the modern Red Sea environment. Below it, the signature changed dramatically. The layer below the boundary contained minerals and isotope ratios consistent with an environment much older than the Red Sea in its current form. Some researchers interpreted this as sediment from the period before the most recent major flooding of the basin, when the area was either a different kind of sea or a drying basin with very different conditions. Other cores from different pool locations revealed layers with high concentrations of organic material that had no clear source in the modern Red Sea ecosystem. The organic compounds were chemically different from anything currently living in the region. Some of them match compounds found in ancient microbial mat communities, the kind of dense bacterial ecosystems that covered large areas of the early Earth's shallow seas. If those interpretations are correct, the cores are capturing traces of life from an environment that existed before the modern Red Sea. The brine preserved them through multiple geological transitions, through the drying of the basin and its refilling, through millions of years of change above. A separate set of cores from a pool near the Kibrit Deep showed a dramatic mineral shift at a depth corresponding to a period roughly 5 million years ago. The mineral composition below that layer suggested conditions consistent with an evaporating shallow sea, exactly what scientists believe existed in this basin before the modern Red Sea formed. The brine pool floors are keeping records of geological events that erase themselves everywhere else. Scientists are now drilling longer cores from the deepest and most chemically stable pools. The goal is to reach sediment from the original evaporation event itself, the moment millions of years ago when the ancient sea dried and left the salt deposits that would eventually create the brine pool sitting above them today. Getting to that layer would close a loop in the geological story that has been open for decades. But while scientists are drilling down into the past, a separate and very modern pressure is building above these pools. Scientists studying the deepest and oldest sediment layers from Red Sea brine pools have been pushing an idea that started as a hypothesis and has slowly gathered evidence. The idea is this: the brine pool environments of the Red Sea, or environments very similar to them, may have existed during a critical window in the history of life on Earth, and they may have been the locations where some of the earliest complex cellular life first took hold. Complex life, meaning cells with a nucleus, the type of cells that eventually became every plant, animal and fungus on Earth, appeared in the fossil record somewhere between 1.5 and 2 billion years ago. Scientists know it happened. They do not fully know where or how the first nucleated cells arose. One leading theory proposes that the jump to complexity happened in environments where the chemistry was extreme enough to drive rapid molecular innovation, but stable enough to protect fragile new cellular structures. Brine pool environments fit both conditions. The chemistry is extreme, pushing organisms to develop new molecular solutions. The stability of the brine means those new structures are protected from the physical disturbances that would destroy them in normal ocean environments. The chemical signatures found in some deep Red Sea brine pool sediment layers include lipid compounds, which are fatty molecules that form the membranes of cells. Some of those lipids have molecular architectures that are associated with ancient archaea and early eukaryotic cells, the first cells that had a nucleus. Finding these compounds in brine pool sediment does not prove that complex life evolved there. It shows that the chemical conditions supporting early complex cellular life were present in those environments, and that the products of that life were preserved in the brine. But it raises the question seriously enough that several research groups are now targeting older brine pool sediment, specifically to look for biomarkers, the chemical fingerprints of life, that date to the billion-year timescale. If biomarkers consistent with early complex life are found in the deep layers of Red Sea brine pool sediment, it would support the idea that extreme brine environments played a role in one of the most important transitions in the history of life on this planet. The implications would extend beyond Earth immediately. Europa and Enceladus both have conditions consistent with brine pool environments on their sea floors. If brine pools helped produce complex life on Earth, the same process could be operating in those oceans right now. Scientists are not claiming the Red Sea brine pools created all life. They are saying the evidence pointing toward them as candidates for major early life events is stronger than it was 20 years ago. And every new core pulled from those floors adds more data to an argument that is still being made. The Red Sea Rift is not slowing down. The two tectonic plates pulling Africa and Arabia apart are still moving, still widening the gap between them, still thinning the crust and letting more heat rise from below. The rift is widening at roughly an inch per year, which sounds small until you remember it has been going at this rate for millions of years. As the rift widens, new cracks open in the seafloor. Those cracks give seawater new pathways to reach the ancient salt deposits below. More seawater dissolving more salt means more brine forming and flowing into the existing pools and potentially filling new depressions that have not yet become pools. The Atlantis 2 Deep, already one of the largest brine systems in the Red Sea, has been measured multiple times over several decades. Volume estimates from different time periods suggest the pool is slowly increasing in size. The rate is not dramatic enough to be visible on a human timescale, but it is measurable. Some of the growth comes from fresh brine input seeping in from below. Some comes from the walls of the pool slowly dissolving, as the brine chemistry attacks the surrounding rock and sediment. The minerals from that dissolution add to the pool's chemical load, gradually concentrating it further. New vent activity in the region can also shift where brine pools form. A vent that opens in a new location heats surrounding sediment, accelerates dissolution of nearby salt deposits, and can initiate brine pool formation in a depression that had none previously. Scientists monitoring the Red Sea floor over long periods have noted changes in the locations of active seepage zones, areas where brine is actively rising through the seafloor. Some seepage zones that were active in earlier surveys have quieted. Others have become more active. The system is dynamic, responding to ongoing geological changes below. The chemical concentration of some pools has also shifted between surveys. A pool measured in the 1990s and remeasured in the 2000s showed higher concentrations of certain dissolved metals, suggesting increased vent input over that period. The pool was becoming more extreme, not stabilizing. This dynamic nature has practical implications for any human activity planned near or above these pools. An industrial operation that assumed a brine pool was static and safely located away from a work area could find the pool has shifted or expanded by the time the operation reaches that depth. And there is now serious planning for industrial operations on the Red Sea floor. The reason is sitting in the brine itself. In the 1960s, before the full extent of the brine pool system was known, a research ship was collecting water samples from the central Red Sea at depths of around a mile. The temperature sensors on the sampling bottles started reading values that made the scientists check their equipment twice. The water at that depth was supposed to be cold. Deep ocean water, away from surface heating, stays near freezing. But the readings showed temperatures far above what any open ocean depth at that location should produce. When the samples came up, the water inside the bottles was visibly different from normal seawater. Darker, more viscous. And when the chemical analysis ran, the team saw numbers for dissolved metals that looked like a laboratory accident had contaminated the sample. Iron, zinc, copper, and manganese were present at concentrations that exceeded anything previously recorded in natural seawater by orders of magnitude. The ratio of certain isotopes in the sample ruled out contamination. The signal was real. The team had stumbled into the brine pool system that would later be named the Atlantis 2 Deep. And the metallic concentrations they measured became one of the most dramatic findings in the history of marine geology. Those metal concentrations exist because hydrothermal vents have been pumping mineral-rich water into the pool continuously for thousands of years. The metals dissolve out of the hot rock below, ride the vent water up into the pool, and then settle into the sediment as the water cools. Over millennia, the metals accumulate in the sediment at the pool floor in quantities that concentrate them far above what the surrounding rock contains. The sediment at the bottom of the Atlantis Too Deep is estimated to contain zinc, copper, silver, gold and other metals in quantities that would be commercially significant if they were extracted from a land-based mine. Scientists calculated the total metal content of the Atlantis Too Deep sediment layer in studies published in the 1980s. The numbers put the estimated value of those deposits in the billions of dollars. That calculation was not made in a vacuum. It was made in an era when deep-sea mining was beginning to look technically feasible for the first time, and the governments of the nations bordering the Red Sea were paying close attention. The Atlantis Too Deep sits in international waters in the central Red Sea, which means its resources fall under the jurisdiction of the two nations closest to it, Saudi Arabia and Sudan. In the 1970s, the two countries formed a joint commission specifically to manage and potentially exploit the mineral resources of this brine pool region. The commission commissioned engineering studies on whether the metal-rich sediment at the bottom of the pool could be extracted economically. The results were promising enough to attract interest from mining companies. The metalliferous muds at the bottom of the Atlantis Too Deep contain zinc, copper, silver, and gold at concentrations significantly higher than average ore grades mined on land. Zinc alone, based on some estimates, is present in quantities that could supply a major industrial user for years. Deep-sea mining companies have developed technology designed specifically for metal-rich seafloor sediments. The general approach involves sending a pump system to the seafloor, vacuuming up the soft sediment layer where the metals are concentrated, lifting the slurry to a surface ship for processing, and returning the depleted sediment to the seafloor. The challenge in brine pools is the brine itself. The equipment has to operate inside a liquid that corrodes metal rapidly, destroys seals and gaskets, and makes operating conditions unpredictable. The density of the brine means pumping sediment from the pool floor requires more energy than pumping from a normal seafloor. The helicline creates operational complications for vehicles crossing between the brine and the ocean above. Several feasibility studies have concluded that extraction is technically possible, but economically sensitive to the price of the metals being recovered. When zinc and copper prices are high, the deposits look commercially attractive. When prices drop, the economics become less favourable. A trial extraction was conducted in the Atlantis Too Deep in the 1980s as a proof of concept. Sediment was successfully lifted from the pool floor, brought to the surface, and processed. The metals were recovered at the predicted concentrations. The trial was not scaled up commercially at that time, largely due to falling metal prices, but the deposits are still there. The technology has improved dramatically since the 1980s. Metal prices have risen, and the interest from mining companies has returned. Every scientist who has studied the Red Sea brine pools for their biological or geological value knows what a mining operation would do to those ecosystems. The extraction process for metalliferous muds works by physically removing the sediment layer from the pool floor. That sediment layer is the archive. It is the undisturbed record of thousands of years of climate history. It is the habitat for the microbial communities that scientists are just beginning to understand. It is the location of the novel compounds that pharmaceutical researchers are working to develop into new treatments. Removing it would destroy all of that simultaneously. The sediment slurry pumped to the surface would also need to be returned somewhere. Deep-sea mining operations typically discharge the processed, depleted sediment back into the water column at depth. In a brine pool region, that discharge would alter the chemistry of the surrounding area. Particles from the disturbed sediment would drift into adjacent pools, crossing their haliclines and potentially changing the chemistry of pools that were previously undisturbed. Scientists who have argued against mining the Atlantis Too Deep and similar pools have pointed to the unique and irreplaceable nature of these environments. The microbial communities inside them have evolved in isolation for thousands of years. Destroying their habitat eliminates organisms that may produce the next generation of antibiotics, or that hold biological information about the early history of life on Earth, before anyone has had the chance to study them fully. The counter-argument from mining interests focuses on the economic significance of the resources and the fact that the metal deposits were formed by geological processes that will continue to operate, potentially regenerating the deposits over thousands of years. Scientists reject the regeneration argument. The timescale of sediment recovery is thousands of years. The timescale of the biological communities that depend on that sediment is also thousands of years. A mining operation that removes the sediment today does not produce a recoverable ecosystem within any human time frame. International bodies dealing with deep-sea resources have not yet established clear legal frameworks specifically for brine pool ecosystems. The question of whether brine pools qualify for special protection under existing marine environmental law is still debated. Several research institutions have formally petitioned for the establishment of protected zones around the major Red Sea brine pools, arguing that their scientific and pharmaceutical value far exceeds their value as metal deposits, especially given advances in land-based recycling of critical metals. The debate is unresolved. The pools are still open. The word irreversible is used carefully in science. Most processes reverse on some timescale. Forests regrow, glaciers reform. Even species driven to extinction leave genetic information that cloning technology may one day recover. The loss of an undisturbed brine pool sediment record is different. The laminated sediment layers in a brine pool took thousands of years to form in conditions that cannot be recreated. The specific sequence of climate events, biological inputs, volcanic episodes, and chemical changes that created each layer happened once. They are recorded in those layers and nowhere else in the same detail. If a mining operation removes those layers, the record is gone. No technology can reconstruct it. No amount of sampling before extraction can preserve a dataset equivalent to the intact archive. Sampling captures what scientists know to look for. The intact archive preserves everything, including what scientists have not yet thought to look for. The biological loss is equally permanent. The microbial communities living in the Red Sea brine pools have spent thousands of years adapting to conditions that exist nowhere else on Earth. Their evolution is specific to their pool. Their chemistry reflects a history of competition, resource access, and environmental pressure that is unique to that location. Destroying the pool does not just kill the organisms currently living there, it erases the evolutionary product of thousands of generations of adaptation. The genome of a brine pool microbe is a library written over millennia. That library does not get rewritten if you destroy its only copy. Scientists studying these pools have repeatedly emphasized that the catalogue of organisms and compounds present in Red Sea brine pools is far from complete. Research has been ongoing for decades, but the pools are large, the conditions are challenging, and the diversity of microbial life inside them is extraordinary. Current estimates suggest scientists have characterized only a small fraction of the organisms living in the major Red Sea brine pools. Many of the remaining organisms may produce compounds with medical, industrial, or scientific value that has not yet been identified. The pharmaceutical industry's interest in brine pool microbes is based on a very small number of screened organisms. Expanding that screen to the full biological diversity of the pools would require years of additional sampling and analysis. That work is in progress, but its completion depends entirely on the pools remaining intact. The case for preservation is not just ecological sentiment, it is a calculation about which use of these resources produces more value over a longer time frame. Metal deposits can be replaced through mining elsewhere or through recycling. A 4,000-year sediment archive cannot. Every ocean world in the solar system that scientists consider a candidate for life shares features with the Red Sea brine pools. Europa's ocean has no sunlight. Its floor is in contact with silicate rock that releases minerals into the water. Gravitational forces from Jupiter drive internal heating, possibly creating hydrothermal activity on the seafloor. The water is salty, the chemistry is extreme. Enceladus has active vents that push water, salts, and organic molecules through its ice shell into space. The chemistry of those plumes matches what you would expect from a seafloor ocean interacting with hot silicate rock. Hydrogen in the plumes points to active serpentinization reactions. the same kind of chemistry that feeds the base of some deep-sea ecosystems on Earth. Titan, Saturn's largest moon, has lakes and seas, but they are filled with liquid methane rather than water. The chemistry is so alien that even the organisms that might live there, if any exist, would be unrecognizable to biology as we know it. Of all the candidate environments for extraterrestrial life, Europa and Enceladus are the ones that most closely resemble what the Red Sea brine pools represent: liquid water, darkness, extreme chemistry, mineral-rich environments, and the possibility of chemosynthesis as an energy source. Scientists studying the Red Sea brine pools are doing something that extends beyond marine biology. They are building a template. They are establishing what the chemistry, biology, and physics of a brine-based deep-sea ecosystem look like from the outside, so that when spacecraft instruments sample the plumes of Enceladus, or eventually reach the ocean of Europa, scientists will have a comparison. The biosignatures of a brine-adapted chemosynthetic microbial community, the specific ratios of elements, the kinds of organic molecules produced, the gases released as byproducts, have been mapped in Red Sea brine pools in enough detail to inform the design of instruments meant to detect life on other worlds. The Europa Clipper mission, which launched in 2024, carries instruments designed partly based on what scientists learned about brine chemistry and biology from places like the Atlantis Too Deep. If the mission detects signals consistent with biological activity in Europa's ocean, the reference data from the Red Sea brine pools will be central to interpreting those signals. The lakes beneath the Red Sea are extreme, they are deadly, they are remote, and they may be humanity's best preparation for finding life somewhere else in the solar system. Scientists have been studying the Red Sea brine pools for more than 50 years. They have mapped several of the major pools, collected hundreds of sediment cores, cultured dozens of microbial species, analysed the chemistry of multiple brine layers, and documented ecosystems that nobody expected to find. And the list of open questions has grown longer, not shorter. No one knows exactly how many brine pools exist in the Red Sea. The deepest and most remote sections of the Central Rift have not been fully surveyed. The technology for deep brine pool exploration has improved substantially in recent years, but a complete inventory of every pool in the Red Sea system does not exist. New pools are still being found on each survey cruise. The origin of some organic compounds found in the deepest sediment layers remains unexplained. A handful of molecules identified in cores from the Atlantis Too Deep region do not match any known biological or geological source. They may represent the products of microbes that no longer exist in the pools. They may reflect chemical processes that have not yet been characterized. They may point to an origin that would require revising existing models of the Red Sea's geological history. The full genetic diversity of the brine pool microbial communities has not been catalogued. Metagenomic studies have revealed enormous complexity in the biological communities of the major pools, far more species than anyone expected. But the databases those studies compare against are incomplete for extremophile organisms, meaning many of the genetic sequences identified in brine pool samples do not match any known species. An unknown number of organisms living in these pools have never been characterized. The mechanism by which certain brine pool microbes resist the toxic effects of the dissolved heavy metals in their environment is not fully understood. The concentrations of zinc, copper and other metals inside pools near active vents are high enough that they should be lethal even to organisms evolved for extreme conditions. Some species survive them anyway. The molecular machinery they use to manage metal toxicity has been partially described but not fully worked out, and the deepest question remains the most provocative. Whether the brine pools contributed to the origin or early diversification of complex life on Earth is still an open investigation. The chemical evidence is suggestive. The biological evidence from the microbial communities is consistent with the hypothesis. But direct evidence connecting brine pool chemistry to specific evolutionary events in the history of life has not yet been established. The lakes beneath the Red Sea have answered more than most people imagined possible when the first scientist watched a camera pass through an impossible surface 50 years ago. They have revealed that life can survive anywhere, chemistry provides energy and water stays liquid. They have shown that the deep sea floor is not an empty desert but an active, complex, diverse world. What they have kept hidden so far may be the most important part of all.